Dear student, welcome to the Medical Genetic course. Today, we will talk about the pattern of inheritance non-Mendelian Part 2. In this lecture, we will discuss Part 2 of non-Mendelian inheritance, which include mosaicism and mitochondrial inheritance. We will start with defining mosaicism. Mosaicism is a condition in which an individual or a particular tissue of the body consists of more than one cell line that differ genetically throughout an error occurring during mitosis at any stage after zygote formation. But the question is, when does mosaicism occur? It happened during mitosis at any stage after conception. Mosaicism can also exist as a new mutation arise in somatic or early germ cell division. Mosaicism of either somatic tissue or germ cells can account for some example of unusual pattern of inheritance. Mosaicism could be in somatic cells or germ cells. In this slide, we will talk about somatic mosaicism. When does it happen and its feature? It could occur after conception or later in life. It is a major contributor to many types of cancer. Also, there is no risk of passing on the mosaic genotype to offspring. Somatic mosaicism of a single gene disorder could be confined to a particular part of the body in a segmental distribution and less severe, such as neurofibromatosis type 1. The variation in skin pigmentation occurring in segmental or pattern distribution, as we see in the picture. Neurofibromatosis type 1 is a common autosomal dominant disorder. Sometimes it is segmental, affecting only one part of the body. Segmental NF1 is caused by mosaicism for a mutation that occurred after conception. Mosaicism also occurs in the germline, which is ovaries or testes, and the rest of somatic cells are normal. If a mutation occurs in one cell of the developing embryo in the germline lineage, then germline mosaicism results. The feature of germline mosaicism: if a mutation occurs in germline, then it will be passed to generation. Usually, it's observed with autosomal dominant and X-linked mutation. Individuals are not aware of their mutation until they passed it on to their generation. Therefore, it's important to keep germline mosaicism in mind when providing reoccurrence risk in genetic counseling for apparently new autosomal dominant and X-linked recessive mutation. So, we have talked about mosaicism and how it affects the pattern of inheritance. Now, we will talk about mitochondrial disorder. As we know, the great majority of genetic diseases are caused by alteration in nuclear genome. However, a small but significant number of diseases can be caused by mutation in mitochondrial DNA. The human mitochondrial genome is extremely small compared with the nuclear genome, and mitochondrial genetics present unique clinical and experimental challenge. Mitochondrial DNA mutations are an important cause of inherited disease. We will talk about basic mitochondrial genetics and the relationship between inherited mutation and disease phenotype. What is mitochondrial DNA? Mitochondria are found in all nucleated cells. Mitochondria are important for cell survival. Mitochondrial genes encode proteins that participate in protein synthesis and energy production. Mitochondrial DNA consists of total of 37 proteins, including 2 ribosomal RNAs, 22 transfer RNA, and 13 essential polypeptides that function in cellular respiration and the synthesis of ATP, which is the biological energy molecule. Mitochondrial DNA mutate faster than nuclear DNA for 2 reasons. It has fewer ways to repair DNA. It is the site of energy reaction that produce oxygen-free radical that damage DNA. Mitochondrial DNA is not wrapped in proteins. Its genes don't have introns, but mitochondria has several copies of its chromosome. At cell division, the multiple copies of mitochondrial DNA in a cell replicate and sort randomly in newly synthesized mitochondria. The mitochondria is distributed randomly between the two daughter cells. This process is known as replicative segregation. Mitochondria that contains only a pure population of normal mitochondrial DNA or a pure population of mutant or normal mitochondrial DNA is called homoplasmy. When a cell receives a mixture of mitochondrial DNA, mutant and normal, some without mutation, it's called heteroplasmy. And the severity of the disease depends on the number of mitochondria in the cell. Did you know that we inherit our mitochondrial DNA from our mother? Because at fertilization, all mitochondria are contributed by the oocyte and transmitted throughout the maternal line. Fathers with mitochondrial DNA mutation are at no risk of transmitting the defect to their offspring because the sperm does not have mitochondria in the cell. The mode of inheritance in mitochondrial disease has features. It includes that all children of mothers who have homoplasmic mutation in mitochondria will inherit the disease. Mutation in mitochondrial DNA affect both males and females. It's very important to know that mitochondrial DNA is inherited exclusively through the maternal line. The disease may be mild, moderate, or severe according to the distribution and amount of abnormal mitochondria in the affected cells and tissues. Expressivity may vary widely among siblings, depending upon how many mutation-bearing mitochondria were in the oocyte that develops into an offspring. More than 100 different rearrangement and 100 different point mutation have been identified in mitochondria DNA that can cause human diseases. The organ systems with large ATP requirements tend to be the most seriously affected by mitochondrial diseases, such as central nervous system, skeletal muscle, and heart. Some examples of mitochondrial disease including myoclonic epilepsy and rigid red fiber disease, and mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes. Lastly, these are some references for the lecture. At the end, we have covered the unusual pattern of inheritance, including mosaicism and mitochondrial inheritance.